Now come to the next question, that is lecithin, right? Lecithin is, what is that lecithin? We'll study it in the chapter of in contents of the liver, that is the bile, right? That is the function of the liver, secretion and synthesis of bile, because lecithin is present over there also. Lecithin acts as a surfactant in the at the surface of the Clara cells, right? In the lungs. So what is that lecithin? Lecithin is a type of the phospholipid actually, right? Lecithin is a type of a phospholipid. It's not a type of wax, not oil, not simple fatty acids. So what is lecithin? It's just a fat that is essential in the cells of the body, right? It is a fat that is essential in the cells of the body. It can be found in many foods, right, including the soya beans and the egg yolks, right? That means that lecithin taken as a medicine as well, and used in the manufacturing of the medicines, right? It is used for treating the mental disorders such as the dementia and the Alzheimer's, and we know that dementia, that is the loss of memory, right? And Alzheimer's is a type of that dementia, right? It is the type of that short term memory loss, Alzheimer's disease, all they are types of the dementias, right? So lecithin is also called the phosphatidyl choline, any one of the group of phospholipids, right? So the answer is lecithin is a phospholipid, that is phosphoglycerides, that are important in the structure of cell and their metabolisms, right? So the pure lecithin is a white, waxy, pure lecithin is white, waxy and darkens when exposed to the air, right? So that is all about the lecithin. Now come to the next. Lipids that insulates the nerve fibers. We know that about the nerve fibers, these are the structures which are present in the nervous system, right, which is the important part of the nervous system. Lipids are not the lecithin. Lecithin is the phospholipid, right? Cholesterol, not exactly, but all are the type of that, but when we talk about the insulation of the nerve fiber, right, so the answer is lipids that insulates the nerve fibers, they are the glycolipids actually. The type of the lipids which insulate the nerve fibers called the glycolipids, right? So what are these? See, when we talk about the cholesterol is a lipid but with a unique structure, right, which consists of 4 linked hydrocarbons ring, right, which consists of 4 linked hydrocarbon rings, which forms the bulky steroid structure, right, that forms the bulky steroid structure. And there is a hydrocarbon tail which is linked to one end of the steroid, and a hydroxyl group linked to the another end, right? That is the structure of the cholesterol. That hydroxyl group is able to form the hydrogen bonds with nearby carbonyl oxygen, right, of the phospholipid. So simply that cholesterol is also known as the sterol. Why? Because it is made up of the alcohol. It is made up of that alcohol and the steroid, right? That cholesterol is present in most animal membranes, because we know that the cholesterol helps in the synthesis of the biomembranes, the steroidal hormones, right, the vitamin D, all these things are formed by the help of the cholesterol. That's why the little bit amount of cholesterol is good, but if it is in excess amount, then it will be harmful for our body, right? So these are present in the animal membranes, right, varying amount, but it is absent in the prokaryotes, right? Cholesterol is absent in the prokaryotes and the intracellular membranes. Clear? So that is a short summary of the cholesterol. Now come to the suberin. As its name suggests, that suberin is the lipophilic macromolecule, right, which is found in the specifically plant cell wall, right, which is found specialized plant cell walls. Whenever the insulation or protection towards the surrounding is needed, right, wherever the insulation and protection is needed, where there is suberin is present here, right, and suberized cells form the periderm, right, these suberized cells form the periderm. You know that suberin, you will study it in the portion of botany, right, that tissue envelopes that the secondary stems as a part of the bark and develop as a sealing tissue, right, after wound or the leaf abscission, right. So that suberin is the lipophilic macromolecule. When we talk about the next, that is glycolipids, right, why lipids which insulates the nerve fibers are the glycolipids. These are the lipids with the carbohydrate attached by a glycosidic covalent bond, right. So this, their role is to maintain the stability of the cell membrane, right, and facilitate the cellular recognition, and that's why that is the actual that's why it helps to insulate the nerve cells, right, which is the crucial to the immune response and in connections that allow cells to connect to one another, right, connect to each other to form a tissue. And as we know that the tissue, that is the group of the cells, right. So that's why the lipids that insulates the nerve fibers are called the glycolipids, right. Come to the next, the pentose sugar which are present in the RNA, right, that RNA, what is the full form of that RNA? That is ribose nucleic acid, right. What is the full form of RNA? That is ribonucleic acid, right. So simply see that the pentose sugar present in the ribose, as its name suggests, ribonucleic acid or ribose nucleic acid, right. So what are these? The galactose, the sucrose and the fructose, right. As we know that fructose is the fruit sugar, clear. See that galactose is the disaccharide that is made up of two sugar molecules, that is the galactose and the glucose, right, that bound to each other. Sucrose is the common sugar, which is a type of disaccharide, a molecule which is composed of the two monosaccharides, that is glucose and fructose, right. That sucrose is made up of glucose and the fructose, and that sucrose is produced naturally in the plants, clear, from which to table sugar is refined, right. That table sugar is refined with the by the sucrose, right, and it has the molecular formula C12H22O11, clear. Come to the ribose. What is that ribose? It is simple sugar actually, right, and the carbohydrate with molecular formula, that is C5H10O5, right. The linear composition is HCHOH whole 4 and H, that is the naturally occurring form. D-ribose is a component of the ribonucleotides from which the RNA is formed, right. And what will happen? So that's why that compound is necessary for the coding, the decoding, the regulation and the expression of gene. That's why we say that the RNA and DNA both are acts as a genetic material, right, just because of the coding, decoding, the regulation and the expression of the genes. And in detail about the RNA, DNA and the nucleic acids, you will study under the in the chapter of the botany, right. Come to the next, that is fructose. Fructose is the fruit sugar, which is a simple ketogenic monosaccharide, right. Learn that It is simple ketogenic monosaccharide which is found in many plants, right, where it is often bounded to the glucose to form the disaccharide sucrose, right. So it is one of the three dietary monosaccharides. It is one of the three dietary monosaccharides along with the glucose and the galactose, right, along with the glucose and the galactose, and that are absorbed directly into the blood during the digestion, right. As we know that when the digestion of the carbohydrate is completed in the small intestine, right, by the help of the disaccharidases, right, and these disaccharidases are the sucrase, the maltase and the lactase. The sucrase is also known as the invertase, which acts upon the sucrose and convert it into the glucose and fructose, right. Maltose is catalyzed by the maltase enzyme and converted into two molecules of the glucose, and lactose is acted by the lactase enzyme and converted into the glucose and galactose, right. So that's why they are absorbed directly into the blood during the process of digestion, right. Come to the next. Nucleoside is, what is nucleoside? Nucleoside is the combination of the sugar and the nitrogenous base, the sugar and the phosphate, the nitrogenous base and the phosphate, or the purine and the pyrimidine. So we know that nucleoside is the combination of the sugar and the nitrogenous base, right. That nucleoside consists of simply a nucleobase, which is also termed as the nitrogenous base, right, and a 5 carbon sugar, that is ribose, clear. So whereas a nucleotide, when we talk about the nucleotide, then what will happen? Nucleotide is composed of a nucleobase and a 5 carbon of sugar, and one more phosphate group is there, right. So in nucleoside only sugar and nitrogenous base is there. In nucleotide, sugar plus nitrogenous base plus the phosphate group, right. So the answer is the A. In bDNA, one full turn of the helical strand contains, right. See, first we have to know about the bDNA. What is that bDNA? That bDNA is the B form of DNA, which is the right-handed double helix, right, which is the right-handed double helix, which was discovered by the Watson and Crick, which is based on the X-ray diffraction patterns, right. Actually, it is the common form of the DNA which exists under the normal physiological conditions. This double strands of bDNA runs in opposite direction, and you will study it in detail in that chapter nucleic acids, right, in section of botany. That in bDNA, the most common double helical structure which is found in the nature, that double helix is right-handed with about 10 to 10.5 base pairs per turn, right. So according to that data, the correct answer is the 10 base pairs, right. That is the 10 base pairs, because we have the minimum range, that is the 10 or 10.5. So that's why we mark the answer 10 base pairs, not 11, right, clear. So the answer is 3. Come to the next question. In a DNA molecule, adenine of one strand base pair with dash on another strand. So first, when we have to knowledge about the little bit about the DNA molecule, right. So when we talk about the chemical structure of DNA, see that is the sugar phosphate, which is called the backbone, right, which is called the backbone. Actually, DNA is a polymer which is made up of the small units, which is called the nucleotides, right, which is called the nucleotides. And these nucleotides are made up of 3 different components. That is, first one is the sugar group, second one is the phosphate group, and third one is the bases, right. These are the nitrogenous bases, clear. And there are 4 different types of bases there, that is the adenine, thymine, guanine and the cytosine, right? So that is the structure of adenine, that is the thymine, that is the guanine, and that is the cytosine, right? So what holds the DNA strands together? See that is the double helical structure of the DNA, and these are the base pairs which are attached with each other by some bonds, right? The DNA strands are held together by the hydrogen bonds, right, between the bases on the adjacent strands, right? You can see here these are the hydrogen bonds which are present on the in between the bases on the different strands, right? Remember, always adenine always pairs with the thymine, right, while guanine always pairs with the cytosine. Clear? Adenine always pairs with the thymine, and guanine also pairs with the cytosine, right? And the adenine pairs with the uracil instead of the thymine in RNA, right? Clear? So that's why hydrogen bonds attach these nitrogenous base with each other on adjacent strands, right? From the basis of single stranded DNA acts as a code, right? The basis of single stranded DNA acts as a code, and the letters from 3-letter codons, right, and which codes the amino acids, the building blocks of the protein, right? DNA to RNA, that is transcription, and then translation, that is the from RNA, that for formation of the protein, that is translation, right? So for that process an enzyme is necessary, that is RNA polymerase, which transcribes the DNA into mRNA, that is the messenger RNA, right? That is the messenger RNA. It splits apart two strands that form the double helical structure, right? Then reads a strand and copy the sequence of the nucleotides, right? So in that pattern, that is the structure of DNA. Clear? Right? A binds with, always paired with the thymidine. C always guanine is always paired with the cytosine, right? A by double bond, G by the triple bond, combines with the C, right? So in that manner, when we talk about the adenine, so adenine is adenine of one strand base pair is combined with the thymine on the another strand, right? So the answer is 2. Come to the next. In bDNA, the rise per base pair would be 3.4 Armstrong, right? So you just have to learn it. Next question. The nitrogenous base of two strands of DNA are joined by the hydrogen bond. Just we'll discuss it in question number 18, right? And we already discussed about the phosphodiester bond, the glycosidic bonds, and the peptide bonds in the previous question, right? So come to the next, that is the genetic material of tobacco mosaic virus is, right? The genetic material of tobacco mosaic virus is the single stranded RNA. So the answer is 1, right? That tobacco mosaic virus, that is TMV genome, consists of 6.3 to 6.5 kb single stranded RNA, right? And the 3 terminus has tRNA, that is transfer RNA like structure, right? And the 5 terminus has the methylated nucleotide capsule, right? So the genome which encodes the 4 open reading frames, 2 of which produces a single protein due to the ribosomal read, right, through a leaky UAG stop codon. So about all these things you will study under the botany section, right? So just in small, I'll just give you a small summary about that, right? Little bit information about that. So the genetic material of tobacco mosaic virus is the single stranded RNA, right? Come to the next question. The double helix model of DNA was proposed by the Watson and Crick, right? Come to the next. In the fifth end of a DNA molecule, the fifth carbon of pyrimidine bases is free? No. The fifth carbon of purine bases is free? No. The fifth carbon of pentose sugar is free? Correct, right? Both 1 and 3? No. So the answer is 3, right? Come to the next. The pitch of bDNA is, that is 34 Armstrong, right? Both of that data is different, right? That is talking about the pitch of bDNA, and in that question, right, in bDNA the rise per base pairs it would be 3.5 Armstrong, right? So both are different thing, don't be confused on that, right? Come to the next. The primary precursor for the production of cholesterol in our body is acetic acid. We will discuss it in the chapter of digestion, right? We already we studied that. See here, what will happen in liver? First formation is the acetate, right? And it is also related with the reproductive system also, right? Acetate, that is acetic acid, right? The primary precursor of the production of the cholesterol in our body is acetic acid. In liver, first acetate is formed. By the acetate, what is formed here? The cholesterol, right? By the cholesterol, formation of the pregnenolone. Pregnenolone forms the progesterone. Progesterone helps in the formation of the testosterone, and it helps in the formation of the estrogen, right? So this is the series, right? That is the series in our body, and that's why we say that the balanced amount of cholesterol is necessary for our body, because it helps in the formation of the steroidal hormones as well, right? So the answer is the acetic acid. Come to the next question, that is metabolic flux is regulated by, right? So the first we have to know that what is that metabolic flux, right? As we it's that word shows its meaning, that is metabolic, that is related with the metabolism which takes participate to complete the metabolic activities, right? And we can say that flux or the metabolic flux is the rate of turnover of the molecules through a metabolic pathway, right? That is, flux is regulated by the enzymes which are involved in the pathway, right? Within the cells, that regulation of flux is vital for all the metabolic pathways to regulate the pathways activity under the different conditions, right? So that is called the metabolic flux, which is regulated by the enzymes. So the answer is A, or the 1, right? Come to the next. In DNA, uracil is replaced by the thymine, right? In DNA, that uracil is replaced by the thymine. Don't be confused with that thiamine. That thiamine is the name of the vitamin B1, right? Which is responsible for, if the deficiency of thiamine is there, then what will happen? The disease, the beriberi Wernicke's encephalopathy is there, right? So that is different thing, and thymine is different thing. So in DNA, uracil is replaced by the thymine, right? Come to the next. Ribozymes are dash that behaves like enzymes. As we focus on that ribozyme, ribose, that is in that particular, and enzyme, right, that is related with the ribonucleic acid, ribose sugar, right, that is related with the ribonucleic acid, and zymes, that is an enzyme, right? So that ribozymes are the ribonucleic acids that behave like an enzyme. So the answer is Bombay. Come to the next. The most abundant enzyme in the biosphere is, right, the most abundant enzyme. As we know that if they ask about the protein, then the answer was the collagen, right? But when we talk about the most abundant enzyme, then the answer is the Rubisco, right? As we know that the collagen is the most abundant form of protein present in the animal's body. Trypsin, that is the proteolytic enzyme, right? That is the proteolytic enzyme which is present in the secretion in inactive state in the secretion of the pancreatic juice, right? And when it active and converted into trypsin, it activates the other enzymes also, right? Insulin, that is the hormone, right, which have the long chain of polypeptide, right, which have the long chain of the polypeptide. 51 amino acids are present in the insulin, and it metabolize the it helps to maintain the carbohydrate metabolized. That means it converted the extra amount of glucose into the glycogen, right? So what is that Rubisco? What is the full form of that Rubisco? Come to here. That Rubisco, the full form of Rubisco is asked in the question in previous year's exam, that is ribulose 1,5 biphosphate carboxylase oxygenase, right? It is the name of the Rubisco. Commonly it is known as the Rubisco, the Rubicase, the Rubco, right? That is an enzyme which is involved in the major step of the carbon fixation, right? That is a process in which the atmospheric carbon dioxide is converted by the plants, right, and the other photosynthetic organisms to energy rich molecules such as the glucose, right? So in chemical terms, it catalyzes the carboxylation of the ribulose 1,5 biphosphate, which is known as the RUBP, right, which is also known as the RUBP, and it is probably the most abundant enzyme on the earth. Clear? When we talk about the fastest enzyme of our body, then it is the carbonic anhydrase, right, of our body, right? Mark my words, of human body, not for the whole universe, right? See here, the trypsin, what is trypsin? It is a serine protease which is found in the digestive system of the many vertebrates, where it hydrolyzes the proteins. That's why I told you that the trypsin is the proteolytic enzyme, right? And that trypsin is formed in the small intestine when its proenzyme form, that is trypsinogen, which is in inactive state, produced by the pancreas, which is activated, right? And trypsin cuts the peptide bond chains mainly at the carboxyl side of the amino acid, like the lysine or the arginine, right? So it is used for the numerous biotechnological processes. Clear? Come to the insulin. As we discussed about the insulin, it is a protein chain or the peptide hormone, right? There are 51 amino acids in an insulin molecules, and the molecular weight is 5808 Dalton, right? Insulin is produced by the cells of the beta cells of the Langerhans, that is the islet of Langerhans of the pancreas, beta cell of the pancreas, right? The name insulin comes from a Latin word, that is insula. Insula means the island, and just because the cell which secretes the insulin is present, is the part of the island of the islet of Langerhans, right? That's why it is called the insulin, right? And that insulin structure varies slightly between the species of the animals. As we know that 51 amino acids are there. There are two chains, right? That is the acidic A chain, and second one is the basic B chain. Acidic A chain carries 21 amino acids, and B chain carries the 30 amino acids, right? And that structure of insulin, chemical structure of insulin, was proposed by or discovered by the Frederick Sanger, and he was the first British scientist who got the Nobel Prize twice in his lifetime within very short duration. For the discovery of insulin, he got the Nobel Prize in 1958, and for the recombinant DNA technology, he got the Nobel Prize in 1982, right? So 1980 or 1982, like that, right? So that is about the insulin. Come to the next question. What is the fate of pyruvic acid under anaerobic conditions in our body, right? As we know that in anaerobic respiration at the time of anaerobic respiration in muscles, the pyruvic acid is converted into the lactic acid through the Cori cycle, right? Muscles in the humans, muscles is able to do the aerobic and as well as anaerobic respiration, right? Muscles perform aerobic as well as anaerobic respiration. In case of anaerobic respiration, that pyruvic acid, right, is converted into the lactic acid. So answer is 3, that pyruvic acid supplies the energy to the living cell, right, through the citric acid cycle, which is also known as the Krebs cycle, right, which is also known as the Krebs cycle. When oxygen is present, that is aerobic respiration. When oxygen is absent, that is called the anaerobic respiration, and in that case it ferments, it degrades to produce the lactic acid, and after that cycle is known as the Cori cycle, right? Through the pyruvic acid is converted into the lactic acid in muscles by the anaerobic respiration. But after the formation of that lactic acid is converted into the glycogen, and that process is known as the glyconeogenesis, right? That glycogen and that process is known as the glyconeogenesis, that is the formation of new molecule of glycogen through the lactic acid, right? So that lactic acid pyruvate is an important chemical compound in biochemistry, obviously, right? Because pyruvic acid supplies the energy to the living cell, right? So this is all about that. Come to the next, that is organic compounds that are tightly bound to apoenzyme, right? Bound to the apoenzyme is. So first we have to know that what is that apoenzyme. So come to that. Apoenzyme, that is an inactivate enzyme, right? The apoenzyme is inactive enzyme. Activation of the enzyme occurs upon binding of the organic or inorganic cofactor, right? The activation of the enzyme depends upon the binding of organic and inorganic cofactor, right? So holoenzyme, that is an apoenzyme together with its cofactor, that is called the holoenzyme, that is whole complete, right? Now in that question, that metal ions is also given, that apoenzyme is cancelled. The answer is prosthetic group, right? That answer is the prosthetic group. And what is that prosthetic group? These are the group of the organic compounds, right? They are tightly bound to an apoenzyme by the covalent and the non-covalent forces, right? Clear? For example, in peroxidase and catalysis, right? Which catalyze the breakdown of, right? So simply come to the metal ion, that is a type of the atom compound that has an electric charge, is called the metal ion. Such atoms willingly lose the electrons in order to form the positive ions called the cations, right? And what is the fourth option? That is sulfanilamide. It is also spelled the sulfanilamide, right? It is the sulfonamide antibacterial drug, right? It is sulfonamide antibacterial drug. Chemically it is an organic compound, right, which consists of the aniline derivatives, right? Aniline derivatives with a sulfonamide group, right? So this is all about that question, right? So the organic compound that are tightly bound to an apoenzyme is the prosthetic group, right? Come to the next question. Bacterial pathogen can be controlled by, as we see here, that is antibacterial drug, that is sulfanilamide, right? So what will happen here? The p-aminobenzoic acid, malonate, sulfanilamide, or all of these. So the answer is 3, right? We already discussed it. Now come to the next, that non-protein part of enzyme is known as we discussed it, that is called the cofactor, right? That is called the cofactor. So what is that cofactor? The cofactor is non-protein chemical compound, right? That is required for the protein biological activity, that is required for the proteins biological activity. Actually, many enzymes are required, right? Many enzymes required cofactors for the functions properly. That these cofactors can be considered the helper molecules, right? These cofactors can be considered the helper molecules that assist the enzymes in their actions, right? And these cofactors, that is, we can say the helper factors, right? Cofactors, that is the helpers. It can be the ions or the organic molecules, right? So or we can say the coenzymes also acts as a cofactors, right? So this is the non-protein part of the enzyme. Come to the next. We already studied about the apoenzyme. What is that apoenzyme? Now what is that inorganic catalyst? See that inorganic catalyst is the catalyst which speed up a wide variety of the reaction, right? Which speed up a wide variety of the reactions. That is, for example, that is vanadium oxide and titanium 3 the chloride are the examples of the inorganic catalyst which enhances the sudden speed of any kind of reactions, right? Come to the next, the active site. That active site is the region of an enzyme, right? That active site is the region of an enzyme where the substrate molecule binds and complete their chemical reactions, right? These active sites consist of the amino acid residues that forms the temporary bonds with the substrate, right? That forms the temporary bonds with the substrate, and the residues that catalyzes a reaction of that substrate is called the catalytic site, right? It's called the catalytic site, right? So this is all about the active sites. Come to the next question. In maltose, the glycosidic bonds is formed in between. You just have to learn that answer, or we know that the maltose, when the maltose is catalyzed by the maltase enzyme, then in that at that time it gives 2 molecules of glucose, right? So the carbon 1 of glucose molecule and carbon 4 of the second glucose molecule, carbon 2 of one molecule and carbon 3 of second glucose molecule. So the answer is the carbon 1 of one glucose molecule and the fourth carbon of the second glucose molecule, right? Come to the next question. The given structure represents a monosaccharide which is known as, actually this is the structure of the ribose, right? This is the structure of the ribose. So you just have to learn it. See that the another structures are glucose is just like that, right? That is C6H12O6. This is the fructose and this is the raffinose, right? So this is the structure of the ribose. Now come to the next question. Chitin, which is present in the exoskeleton of arthropods, is the protein? No. The polysaccharide? Yes. The lipid? No. The derivative monosaccharides? No. Actually, we already studied about that. The chitin is the derivative of the N-acetyl glucosamine, right? And it is a type of the polysaccharide. So the answer is 2. The given amino acid dash in nature, as we know that the H is there and all this it shows the acidic nature, right? So the answer is 1, right? Clear? Come to the next. that is the structure of protein which gives the three-dimensional view. It is called the tertiary structure actually, right? It is called the tertiary structure. So the tertiary structure of a protein refers to overall three-dimensional structure, right? When we talk about the tertiary structure, then it is all over three-dimensional arrangement of its polypeptide chain in space, right? It is generally stabilized by outside of the polar hydrophilic hydrogen and ionic bond interactions and internal hydrophilic interactions between the non-polar amino acids chain, right? So the answer is 4. See, these are the structure and we already discussed these structure in previous question, right? So you just have learned it. If the structure of protein is three-dimensional, that is the tertiary structure, right? In this manner, the whole biomolecule chapter is covered through these questions, right? Thank you.